So again, thank you all for joining us today. As a reminder, this public webinar series is designed to educate attendees on the scientific ecosystem, and each month we'll have representation from either a data coordinating center, a center of CFDE, or the development from any of the consortia that are also involved. And so with that, I'm going to give a few updates before I hand it over to Noel Burt, who is going to be our presenter today. But just a reminder that this session is recorded and is available on YouTube. So please feel free to share this with your teammates or anyone who might be of interest. You can scan the QR code here. And this will take you to our YouTube landing page, which has different playlists depending on the different webinars and talks or, special sessions that we have. So with that, we're happy to share any of this as well afterwards, in addition to it being on YouTube. Um, as always, if you have any feedback, questions, or you'd like to be added directly to our listserv if you're not already, please email us at help@cfdeknowledge.org. Just giving an update for our upcoming schedule. Next session, we will have Uniprot in April, and you can see all of the different organizations that are going to be joining us throughout 2026. We do have an opening November 20th. If you are available and interested to present from the ecosystem, we'd gladly work with you to schedule time for that. As a note, November typically overlaps with Thanksgiving, so this is just bumped up a week, and then we don't have any sessions in December, as this usually falls on the holiday break as well. So if anyone has any questions about the webinar schedule, please let me know. I'll have more details as we get closer to these events. Now, for some updates for conferences that we have, there's several conferences that are upcoming in this quarter through various organizations as a part of CFDE. So our next upcoming one is the American Physiology, oh my gosh, I can't speak today, Physiology Summit. And this will be April 23rd through 26th in Minneapolis, and Spark will be there. So please keep an eye out for that. They will also be sharing any information about Spark or the ecosystem. So please feel free to stop by there. The training center and Asset Knowledge Center will be at BioIT World in Boston. This is May 19th through 21st. I have a little bit more information about the events that will be going here later on, but we'll be happy to see you. This is in person and virtual. And then we have the Festival of Genomics Biodata and AI Conference in Boston, June 3rd and 4th. We have this fantastic session that will be going on in our Genome Dome, as we typically do, and we'll also have a booth. So please feel free to stop by. Both of these, the booth and the Genome Dome, are going to be right next to each other. So if you're curious and you want to hear some other talks or meet with us there, we'd be happy to do that. So keep an eye out for these conferences if you'll be attending, or if not, these are some that you might want to add to your agenda for upcoming conferences. As I had mentioned, there is going to be several events at the BioIT World Conference in May. And so the one that I really want to highlight here is the Training Center will be hosting their 7th annual BioIT Fair Data Hackathon. And this will actually take place a day before the conference and the first day of the conference. So just to note that for programming, if you're planning on attending and you're thinking about what dates you want to come. This basically has a focus on open source tools, and we'll be looking at fair data principles. So we're very excited to be a part of this to help with the training center as well. So you'll be able to see both of us there. So if you have any questions or concerns about this event, please let me know. There's also some details about how to register for the event and more of which projects will be involved on the BioIT World Conference site. So if you have any questions about that, I can also bring you to those sessions as well. So we're really excited and glad to be a part of this. Another reminder, there is the Decoding the Data Ecosystem podcast, which is hosted by the CFDE Training Center. And basically, this is designed to be a community for anyone who's passionate about omics research or any pressing health challenges. And this is designed for any type of researcher, whether you're just starting out in your career or you're just curious about the future of biology in general. There are insights that this podcast has that'll tell stories through people that are within the ecosystem and also showcasing incredible scientific work that's going on from individuals. So if you are interested, there are several episodes already available online. And you should be able to listen to the podcast anywhere you typically get your podcasts. So take a look at that. And again, if you have any questions or concern about this, please let me know. And then we have an exciting funding opportunity that I'd like to showcase here, which has been announced specifically for pilot projects enhancing utility and usage of common fund data sets. The specific RFA number is here underneath the QR code. If you're curious about the details of how to apply, please feel free to scan the information there. But essentially, there are valuable and widely available data sets that have been put together in the Common Fund. And the goal of this is to basically demonstrate and enhance the utility of the selected common fund data sets. So the idea would be users could generate hypotheses and create discoveries based off of these data sets there. So basically, there's going to be a segment that's surrounding the feedback on the utility of these data resources as well. And so some important information if you're interested. There is, 13 expected awards with 200,000 award maximum. We just wanted to put that out there so you can see there are quite a lot of opportunities. The closing application deadline is June 23rd, so you have a couple months to apply for this. But we wanted to get this out to you as soon as possible if you're interested. And again, if you have any questions, concerns, please let us know. If you are wondering about some of the wording, we can take a look into that as well for you. So we're excited to have this there. Noel, is there anything you wanted to add specifically for this or in addition to?
No, but we should put you have the QR code. But in the QR code, it's also there's access to existing resources that the Common Fund has made available that might help you generate some ideas. Namely, some stuff from the Knowledge Center that I'll talk about later, but also some stuff from the data distillery and the C2M2 model, which are some of the knowledge graphs we've produced. So those are accessible to you to query to kind of get some ideas. So there's also a combination of like what the RFA is all about, what they're expecting, and these are incredibly good mechanisms, frankly. They're great mechanisms that the Common Fund's made available for you to use Common Fund data in your own research. But also to give you some ideas of where to start, there's also a lot of information on that site right there. So check it out. It's a nice opportunity in this time where we're all looking for new ways to fund our research.
Great, thank you. Awesome. And then I have one other event that I'd like to, share with this community specifically. And this is the Common Fund Data Ecosystem Incubator for Pan NIH Resources. And so the reason we're bringing this to you today is we are working on putting together, like I said, this incubator of ideas July 9th and 10th. And this will be held at the Neuroscience Center in the NIH. And essentially, any information that we have for this, it's going to be added to our site, which again, you can see on the QR code here. We do have a room block that's going to be available for any of the attendees at the Bethesda North Marriott and Conference Center, which we will have more details on a little bit later. But this is just to let everyone know that, save the date. We're really excited about this. Just to give some insight into what this session is actually geared towards. We're hoping that this can basically focus on collaboration. And so what we'd like to do is build a foundation for cross NIH resource interaction and that direct collaboration. We're also focusing on some shared themes, which I will mention in a moment, and use those themes to have pilot projects to drive our progress. And our goal is to basically position CFDE as the initiator for this infrastructure and coordination. So as part of that, as I'd mentioned, there are some scientific themes that have come up that we'd like to address. These are not finalized, but things that we're starting to think about in the community. And so the first is gene sets and using them as connectors across resources, several of our resources, build these out or use them in some way on their resource or their data sets that they're providing. So we're eager to see, how can that be applied to other resources and other activities outside of just the Common Fund data ecosystem. We're also, hoping to have some AI-enabled connections that help bridge gap between systems and also focus on knowledge graphs, which we can use as a integration layer essentially. And so as part of this, the way that we can have all of these connections is through our human collaboration. So actually being in person, talking about our science, and working with the community to strive for, success in these thematic areas. And so, as part of that, I just wanted to showcase what we're thinking about the event design. So, the agenda is currently in progress, but essentially, we will have keynote speakers who will share their scientific updates and their thoughts about the ecosystem and beyond. We'll also have panel discussions, so we get to see, several different topics debated by individuals who will be showcasing their thoughts as well. And then we'll also have breakout sessions where we'll have working groups to specifically look at these scientific themes, and we'll have readout sessions to basically bring back to the entire group and share with the community. Because again, as I had mentioned before, we want this cross collaboration through all of these resources and individuals. So we're really excited about this opportunity. Um, if you're interested in participating, or we have several different options for this, as I had mentioned. If you're interested in attending, please feel free to RSVP here. I'm just going to pause for a second because I have several QR codes for RSVPing, and I want people to have a moment to do that. I can also put in the Google Forms where we're basically taking attendance as well. If you're interested, I can put those in the chat as well. Uh,
Yeah, in the chat. I also have at the end of my slides too, Mackenzie, so people have
Perfect.
multiple opportunities.
Okay, great. So then I'll move on to the next one. If you're interested in speaking, this is a separate QR code with a form. And so the idea would be if you want to be a panelist, a speaker, lead breakouts, things like that, we'd love to have you help us with those. And if you're interested in presenting a poster, we're going to do a poster session at some point, most likely the first day, based off of, interest. We can see how many that we have, but please feel free if you want to share this with your team as well. We're more than happy to have that. So I'll just pause there for a moment.
And thinking on that, we'll have both for people interested in speaking. If you want to recommend someone, like it'd be great if you took this because we want to make this like CFD is just the host. We just want to like bring all the NIH resources in our ecosystem together in one place to talk about scientific interoperability. That's really, and Mackenzie explained it beautifully. And so if you have an idea of like, you should talk to this resource, you could also share that through this mechanism as well. Couple things of note, as Mackenzie is probably going to get to potentially, so I might over let's share before she is, but we also will have some stipends to offer people who are going to be presenting posters or giving talks. So it is an opportunity to come. We want the event to be free for everybody who does attend, though. That is very important to know.
Yeah. Nope, that's perfect. I actually forgot to mention that, so thank you for adding that there. And then I'll just pause one more time if you want to take a picture and use the RSVP code here. And again, if you have any questions, concerns, please feel free to reach out to me. As you know, I'll be handling a lot of the RSVPing and any other questions that you may have. And with that, these are all the events that we have upcoming. If you are interested in attending any of them, we're really excited about that. But now we're going to talk about some scientific updates and new opportunities that you can have by using the Knowledge Center. The key being the new workflow that we have, CFDE Reveal, which is what Noelle is going to be spending a decent amount of our time today. So with that, I will hand it over to Noelle. Thanks, everyone.
Thanks, Mackenzie. That was excellent. All right, let me share my screen and make sure you can see. All right. Everyone see me okay?
I
All
was
right.
muted, but I was saying yes.
I was going to get the little Zoom like thing out of my way. Beautiful. So often we host these for the CFD in general, so we don't always get to speak. So it's actually fun to be on the other side of the fence speaking. So for the rest of the time, I'm going to talk to you about something that's kind of exciting to us, particularly given, our background as human geneticists, how we think about and approach resources we built before we joined the CFD. But being part of the CFD has been quite awesome in terms of teaching us a lot, exposing us to an entire new universe, and also testing us in terms of how we actually integrate the immense wealth of resources across the CFD. So this has been really fun. In fact, just recently, actually like 2 days ago, we just had our annual meeting. So it's actually really fun to come off of that meeting. We all got together and talked about ourselves in this ecosystem, but we really wanted to share what the Knowledge Center has been up to in the last couple months with you all as a means to actually expose things about the CFD more broadly to you, because that's really our job at the Knowledge Center. So remind you, Mackenzie's talked about this. Many of you know the Common Fund, but it's just good to anchor you to what the Common Fund data ecosystem is all about. It's an incredibly rich set of resources that all have their own culture, community, incredible data sets across biology, genetics, genomics, physiology, biochemistry, you name it. And it's anchored together by 5 centers, right? We're the Knowledge Center. And you heard a lot about the other centers today, but we all work together to basically make the CFD data and knowledge more accessible to others. That's really our mandate. And that, again, is sort of summarized here. Our job across all of the CFD is to create a functioning ecosystem that makes Common Fund data sets more accessible, useful, and ideally discovery. And that's really the thick of it, right? We want to make it simple for folks to navigate this complex ecosystem. So it was really cool at our last meeting. We all got together, we all talked about our updates, and one of our more recent centers actually had a launch. So I wanted to mention that here because now you have a cloud workspace where you can bring CFDE data into a place. It's free, mind you, which is super cool. It has all these tools and workflows. They're going to come talk probably at a future webinar, but I just wanted to announce that they had a final official launch for their cloud workspace, and that's really exciting. Now all 5 centers are fully functioning, open to the public, and that's pretty exciting. So I wanted to mention that and flag that to you all. And so with that, I thought this was a great quote, and I don't remember who said it. I'm not giving the proper credit, but I know it was one of our NIH colleagues. And I love this because I think it sets up for what I'm going to talk to you about. It says, if CFD is successful during the 5 years of phase 2, more investigators like yourselves will be using Common Fund data and products for novel discoveries and innovative purposes. And I think that's really important because that's really what I want to focus on today. And hopefully that's if we are all doing our jobs as CFD centers, we will enable this for this community. So I just want to put that there. So the Knowledge Center, those of you who haven't been to it, this is what it looks like when you land on it. It's meant to give you just a snapshot of the knowledge across the Common Fund data ecosystem. So I'm going to walk through some pieces of it for you if you haven't seen it, but just gives you an idea of literally how all these resources connect to omics data, to entities. And as you go through them, you actually get snapshots of all of them. So it gives you this beautiful informational landscape. So the way that we approach, which is going to be important for the feature I'm going to demo later, is the way we approach the Knowledge Center is by building off knowledge from different layers, right? Some simple knowledge, which is what are the foundation, what's the purpose of all the CFD projects? Putting that in one place, right? What are the primary findings, right, of each of the DCCs? I think that's really great if you're just sort of someone who's curious about the CFD, you want to know what the primary findings from Glygen were. You may not want to go to 18 different resources, so our job's sort of pulling that all together to orient you such that you can find what you need in those other sites. So that's primary and secondary knowledge. Secondary knowledge. Secondary knowledge is something that we can derive from either a single DCC or another one that may not be necessarily published. So these are custom analyses we've worked together with all the DCCs to do to kind of expose new users to it, right? So there's all this accessible chromatin data in 4DN. We worked with them to connect it to human phenotype because we're geneticists, and now that's available to all of you. That was something that they had never had the time to do and the interest. We worked with them to do it. So it's sort of the coupling and creating more ways to expose knowledge. So that's been fun. Then finally, the most fun part is tertiary knowledge. This is where the CFD reaches outside itself to say, we have these great foundational data sets, but they're only really relevant if you add them to orthogonal data to make them more meaningful. So that's really what I'm talking about today. I'm going to talk about that in the context of human genetics. So when you enter the Knowledge Center, you get a search button where you can basically type in anything. And we have sort of an LLM-assisted single search that allows you to see kind of a snapshot of if you type in your favorite gene, you'll see where we have results about that gene, what Common Fund programs have relevant information about that gene, and actually it's a little snapshot eventually of what Reveal says about that gene. So that's going to be a piece that's growing over time. And as we add features, that functionality improves. Again, I mentioned this before, if you hover over any of the different DCCs, you get a little description so you can decide if you want to pursue it more. Same thing for our centers. We make sure that we expose all of them. And if you go there, you click there, you don't go to a description, you actually go to the actual training center, which is great. So you can kind of move in and out of the ecosystem in kind of one fell swoop. This gives you a sense of that whole overall larging framework. Then, as I mentioned before, there's a carousel, if you will, just on the landing page of all the secondary analysis we've done. So here I'm showing an example of HubMap. HubMap is a resource that aggregates human healthy tissue across all tissue types, makes them accessible and useful to you. What we've done here is created integrated maps and have a quick snapshot if you want to look at your favorite tissue or cell type right there for you. Bang, bang, bang. It's all there. But if you want to go to the data, you go to HubMap. So it's this nice coupling of knowledge versus data. These are some of our tertiary workflows. I want to talk more about these. What I mean by this is these are things that are anchored to CFD data, but we had to use other data to make them accessible. So one of these is like a classic tool, which is like we've aggregated differential expression from all the expression RNA-seq data across the ecosystem, of which there's a lot. GTEx obviously is the anchoring piece, but 14 other DCCs have gene expression. So then there's one systematic analysis of all that there. And then there's two other approaches, which I'll talk a little bit more, which are linking gene sets to human phenotype or to mouse phenotype and vice versa. So those are two key features, but I'm going to talk about them in the context of Reveal in a moment. And then at the bottom of the page, you have all the wonderful information and things that Mackenzie went through. So that's just a walk through the landing page. So one of the tools that we've moved towards is trying to make the experience a little easier. Sounds like it's a little overwhelming, right? So you see all these resources, you're like, where do I go? And you are a scientist. You have your own thoughts and opinions. And we want to bring those thoughts and opinions in biological context to you. I mean, no, we want to bring yours to the resource. You know what I mean? So the new feature I want to talk to you about is called CFD Reveal, a reasoning engine for validating evidence across the landscape. And this landscape being a start of CFDE, but it's also a wider ecosystem of NIH resources. So let me talk about sort of the concept behind that first, and then I'll get into the details. The original version was very simple, allowed you to put a natural language query in, and it uses semantic search to connect that query to search terms, and then it searched our resource and reported some results. It was pretty opaque, but that's what it looked like to begin with. But what is it anchored in? How's it built? Well, it's all anchored to genes. This is the join key for us, and it's a very common language across human biology, right? It's a way for us to share information, very simple. It's a connection commodity across NIH resources, in my opinion. What's great about genes, we know a lot about them, right? They're very clear. We have ideas, they're stable, they're widely adopted. They also can be compressed into information, right? That they are a portable, computable object, and they have biological meaning. So that's a framework for connection. Let's just start with that. Better yet, gene lists are readouts. They're readouts from a host of different places. You get them from GWAS, gene expression, ATAC-seq, many other things, right? They can easily be shared. They have no protective data to them. It's just awesome. They can be stored in very consistent ways, and they represent meaning and context from the origin, right? So that's another piece that's really important. And critical to that activity for gene sets, we work with our collaborators at the DRC because they've done a lot of extensive work to generate gene lists from these CFD programs. So there's an incredibly rich substrate for us to build upon as a knowledge center. So how do I mean do we build upon it? We take those gene sets, of which there are 200,000, over 200,000 gene sets, and we extract them, right? Most of these are from differential expression, mind you. But then we want to give them some more meaning, right? We want to connect these gene sets to human phenotype and pathway, and the gene's the anchor. So that's the context behind what we do. And we get to do that with the beauty of human genetics. And this is because we can use human genetics to essentially clean the gene sets and create a statistical anchor. Because the nice thing about human genetics, they are observations of nature, right? And they assume causality and have human relevance, right? We all know this as human geneticists. Moreover, genetic association enrichments can link gene sets to human traits, right? So if a gene and the gene sets are enriched for GWAS associations, that can link to a gene set of that trait, right? So that's your context. What's great about what we get to do with our other hat is we aggregate and curate human genetics data from all over the world through our association to function knowledge portal, where we have 1,300 traits we've curated. We have the beautiful OrphanNet that really handles curated rare traits. And we have our collaborators at GWAS Catalog who are constantly feeding us GWAS data that we are meta-analyzing and containing. So this gives you a resource of 7,000 traits to anchor these gene sets to. So those are our big substrates, okay? So in comes a wonderful model. Many of you have been on our webinars, have heard about the infamous pigeon. So this is basically a Bayesian model that allows you to create probabilistic inference and stitch these gene sets to human phenotypes, right? So we talked about this before. I'm not going to focus on this today, but say that that's the core method that makes this happen. So putting it all together, you have your gene sets derived from from CFDE data sets. You have your genetic analysis results from over 7,000 traits, run those through Pigeon and allow you to sort of infer mechanisms. But how do you do that? How do you make that useful? And how do you bring your context to that? It's an incredibly big hairball, if you will. So we have that currently available in sort of like very specific terms where you can go to gene set analysis. You can type in your favorite phenotype, for example, and say, what gene sets and phenotypes are linked to this through this pigeon method. Or you could say, I have a human phenotype, mouse phenotype. Let's start with that. And I want to know what's the human phenotype. And I've used gene sets and statistically anchored human genetics data to make that connection. So that's what these features are. But this is not incredibly useful unless you can actually add context. So our job, not just as a center, right, is to represent CFDE data sets, but also to contribute externally to the wider environment around our ecosystem. And the way we think about doing that is like sort of lowering the friction, right? So I talked about the internal thing, which is like the fact that you get to the site and you can see everything about CFD data, but you actually want it to be super simple for you. You actually are like, I have a question. I want you to connect me to the, I don't want to do all the searching and the thinking and wondering how. That's no, thank you. I'll do that once I have an answer. So our job is to sort of simplify your research curve, if you will, right? So we're going to take you a little bit easier to get that proof of concept. That's really what the Knowledge Center is aiming to do, minimizing the cost, improving the benefit to that initial sort of lookup phase. That's kind of what we've done with the CFD Knowledge Center thus far. But this has limited impact unless you can attach meaning, as I've been saying. Context matters. Everybody's frame of reference is different. And if we actually want to make this useful for biology, that frame of reference tends to be very specific in many cases. So can we expose scientific connections across the CFD through your research context? And that's what Reveal is meant to do. So with that, Reveal, just kind of give a high level, I've sort of described it from the perspective of genes building up. Again, sort of to summarize, we take those gene sets, we extract them, we annotate them and embed them in a vector database so they can be queried, run them with trait associations and make them accessible, and then allow a LLM workflow to query over them. So the first idea is to focus on causal mechanisms. But what you'll notice right away is that when we released this, it was very hard for us to basically say, what can you search? What are you getting back? The UI was a little bit difficult to say, what am I seeing? What am I learning? What's the process? What is basically about data? And what is the LMM inferencing over, right? So we really improved the UI to expose process and source. That's really important. We added cluster nodes to the graph, just in summary, and I'll talk about that in more detail. And we have sort of a full knowledge graph that I want to describe to you. That's the backbone behind what you're querying. And what's most important is we benchmarked this. We actually said, if you use this, and if you had this and you produced a paper, if you had this before you produced the paper, could Reveal have helped you to find some of the primary claims in your paper? And I think that's the exciting part that I'll get to shortly. So some of the challenges. So as we started to use this, we noticed when people would type in a query, they'd get a lot of information back, but they were basically the same gene set. So you can see here, you got motor pack, motor pack, motor pack, because my query has a bunch of phenotypes. There's many different phenotypes linked to my query, which is excellent, and they're statistically rigorous. However, there's a bunch of gene sets, and I'm like, how do I parse through them? They're largely overlapping. So we want to make it more simple, but we also want to attach some more biological meaning to it. So with those two things in mind, we also want to figure out how to expose the fact that, like, just by virtue of the creation of these DCCs, they are already kind of connected to mechanisms. So motor pack, your gene sets are often connected to response to exercise, right? So that's going to immediately be useful for folks studying metabolism, for example. Senet, for example, this is about aging. So those gene sets might tell you something about the process of aging. So how do we make sure that that's exposed with how these gene sets come back to you so that you can make connections across, right? So common approach and what was suggested to us was like cluster some genes. And there's many approaches that exist for this. I'm just giving you an example. But some of these are often difficult to interpret and connect to biology because, they may just do things semantically or things like that. So We had an idea to again use our human genetics backbone to try and bolster the meaning to these gene sets. So ideally, mechanism are a means to connect biology, right? What's the mechanism? What's going on? So here, just sort of a little schematic about, in how to interpretable biological mechanisms, right? So there's a true mechanism, and there's a set of genes involved. But if you do an experiment, you get an observed readout, and that is essentially a probability distribution over genes. And so gene sets are essentially approximation of that distribution. So they're like a mimic, essentially, of a true mechanism, right? If you believe that, because there's something that you perturbed, and that's a readout, and it's expressing what's happening, behind the scenes, if you will, in the cell. However, many mechanisms are too much to enumerate, and they're very similar, right? So insulin signaling fat, insulin signaling liver, insulin signaling under lipotoxic liver conditions. They're very similar, but they have some differences to them, but they're difficult to enumerate. So we actually wanted to potentially refine the number of mechanisms reported back to you so you can actually make some sense of it. So it's more about like making it simpler, but anchoring it to human genetics. So this is a method that we have not yet talked about, and Jason will come back in another event and talk about this in more detail and actually describe the math behind it. But from the perspective of the utility of it, Eagle infers mechanisms. You might be noticing a bird thing. We got the pigeon, we got the eagle, more birds to come. Infers mechanism across statistically anchored to a human trait or disease. So bottom line, it allows you to anchor those gene sets to human phenotype based on genetic associations. So what it does, for example, is it labels mechanisms according to the names of each gene set to highly loaded to each mechanism. So if you see here, you see cardiac hypertrophy, you see cardiac fibrosis, you see an enlarged heart. These are all through this factor matrix labeled as heart failure and hypertrophy. So it's a simplification of the gene sets to a specific mechanism area, right? So it allows it to be just organized. But the nice thing about this is that we've had some validation and application of this to the reveal knowledge graph. And what it has shown is that the traits do reveal some factors. The factors they're biologically plausible, and they tend to be distinct. So that's great. So it tells us we feel good about this. So here's what it kind of looks like. It is what we call our genetic background, right? It has a combination of genes, gene sets, mechanisms, and phenotypes. So you're looking at this, and if you're me, you're like, okay, I can see some connections, but this is difficult. But what this means is it's now these gene sets are contextualized in our genetic backbone. But then now you need some way to interact with it. And that's what Reveal now does. So if you go to Reveal and you type in a query, and here I'm saying the query I typed for our example is find a kidney transport mechanism in which renal transporters regulate excretion of a exercise-derived anexogenic metabolite. That's very specific, and we'll get to why that matters in a minute. But that's the query I put in because I wanted to know this because that's part of my research. So here's what happens. The process. The first thing that happens is you're exposed to what happens, which is really great because you want to know what's going to data, what's going to APIs, and so on and so forth, and pulling back actual physical results and statistical evidence. And then what is the LMM reasoning over? So the first thing is the LMM. The query is parsed into phenotypes or mechanisms based on the search and a research context. Then that semantically matching begins to mechanisms. Those are retrieved. And then gene sets with the high loadings, high scores, that read out the mechanisms are retrieved for you. So this is the process that is exposed to you. So you see the steps in front of you, and you can go back and actually literally open those, which is kind of cool. We wanted to do that to sort of express the real, the guts of what's happening. So then you get data, you get your results. So the LMM does this for you right now. I think this is something we want to work with you to get your feedback. It selects which factors are most relevant to your research context for you. And it lists a set of phenotypes. And it kind of automatically checks them, but you can check them over time. We're going to make this a little bit more useful. But it checks these phenotypes that it believes are most connected to your research context. And there's a lot more here. And then you can navigate through them. It tells you why. And it tells you the gene set cluster. So these are the factors right here. So if you click on show more, this is what's pretty exciting. It tells you its work. So the integration of the Reveal Knowledge Graph has now been integrated in the C2M2 model, which is the CFDE's metadata model, which is great because if you're going to have any results, you want to know where we got them and why, right? And are they real? And you want to go say, I don't believe you, I'm going to get them myself. So this is a great example of how we're demonstrating the provenance all the way through what we're showing you here, all the way to the GTEx data that generated that. So now you can go and say, that's the genes that's the actual genetic gene expression data that generated this result for me. So it provides you with like that full context. This is really important for us across the ecosystem. It may not be as important as you're thinking about using this actual resource, but in terms of thinking about the fact that we have all of these data resource centers to aggregate together to try and represent them accurately, this is a lot of work. So thankfully for these methods and these models like C2M2, we can start to connect these things and make meaningful science that can be reproducible and fair and consistent, you know? So that's one mechanism. And then you get the actual results. You want to see the genes that are in the gene set. You get our scores and the basically the functional support that says why we actually include those. So now the cool part, the results. Oh, this just gives you how you can actually click and link directly to the workbench where you can get that actual data and the actual raw files that were used to generate this gene set, which is really cool. So here's what you see. This is what you see in the results tab. So the first version of this, if you've used this before, was not like this at all. You had no idea what was going on. This is actually a beautiful upgrade, in my opinion, because it shows its work and parses it for you based on that graph. So first of all, now you see the part of the graph. So if you like knowledge graphs, you can start to say like, oh, I see how these connect. And so to me, it was like a little opaque at first, but now I start to get it. So the colors mean something, right? The red are relevant phenotypes. The blue are gene sets and the clusters of gene sets that are oriented to that. And then in green are all the gene sets that are linked to that. So this gives you all of that provenance, so how things all fit together. And the top is the rationale and a summary, which is created by the LLM generator. And then you get the top genes. So that's what you get to see, and you get to play with this, and you can click and point and things. But here's what I really like. So you saw that and you're like, all right, great. But does the resource actually reveal scientific support and connection to real results and data? So this is the hard part. And this is where we need help. So we started to do this. So we did some benchmarking. What we did is we took a bunch of papers, 10 in this case, and we did a literature for past papers, and we said, had they had reveal at the time that they were writing up their manuscript, could they have found anything in there to actually support or confirm the main claim in the paper, or add orthogonal evidence through CFD datasets that could have them with another hypothesis or learn about extra genes or traits? So that's the example. So I'm going to walk you through one of them right now, which is the Lee et al. Paper. So for that, it was this paper that's sort of like something that was close to our hearts. We in our other world, we started common metabolic disease. So this is a paper that found that SLC17A1 and A3 transporters mediate renal secretion of a metabolite that suppresses food intake and body weight. Okay. So their claim in this was this, right? So had they had reveal at the time, they might have typed in that very specific query, which is why I did that. So that query was actually from this paper. And we said, had we had reveal at the time, what would reveal have revealed to us? So this is pretty cool. If you type in that query, it actually recapitulates the two top genes from that finding with strong evidence, both in the GWAS and the functional support, the combined, which is pretty cool. And a similar mechanistic hypothesis, which is great. The second part that I love is that it reveals the top genes, but it also includes other hits, which they may have already known, or this would actually give them more information. So it expanded to 3 other genes, plus urinary metabolite and urate phenotypes that weren't implicated in the paper. So additional phenotypes were exposed. So what this showed us with this early review is that the authors had the reveal had reveal at the time of the paper. They could have expanded their solute transport program around 2 other transporters, or at least substantiated their current hypothesis and used CFD data to sort of support this. So we were encouraged by this. And so we went on to look through the other benchmarking, and we found some pretty consistent results across all of these, with the exception, I think, of one. We either found that there was strong communication, strong confirmation, excuse me, which is in the green button, or we had additional things where it actually supported both, where it said like, man, if you found this too, you would have learned this, or you could have used this CFD data set to actually extend your hypothesis. So this is pretty cool. And what's great about this is this gives us people to talk to who we can actually say, hey, can you use this? You're the real scientist. You understand your biology. You understand your context. Could you help us see if this actually would have been a mental workflow you would have followed? So, it's one thing to benchmark this. We're sort of like saying that, this happened and really structuring our query according to the paper's main claim. But had the researcher known what to do with it, would they have arrived at the same conclusion? So we have a lot of work to do, but this is incredibly encouraging to us as a way to really make CFD data sets transformed into a kind of a new way that I don't think they're currently accessible, right? They're accessible now in a way of contextualized to your context through genetic association, so anchoring them to disease, and then bringing in that sort of mechanism component through all the gene sets linked to actual biology. So that's the sort of premise behind Reveal. So what now? Well, we got to contact the authors. We want to, some of them we luckily know, which is great. But really, this is where you all come in. I'm really excited for you to try it. I'm trying it out because I'm a biologist but I don't have a wet lab. I'm not sitting there studying a mechanism or process every day. I'm making resources to help you. So we need biologists to actually type in what they think they would want to know and see what comes back and work with us and tell us nah or yeah or I'd want this. Even more so, there's an opportunity here. We actually have funds in an award to actually support the validation of some of the results from this. So if you do find something and you actually are like, I want to pursue it, please come talk to us because we have the funds to make that happen for you. So that's really important for us because this is actually really a way to actually hopefully makes people's scientific lives easier using all of the common fund resources. So what else, right? So you have this backbone, right? And if you're a biologist, you're like, I want a natural language query. I want to work with it a certain way. But if you're a data scientist, you are extremely excited by this because this allows us to expand our connection in other ways across the ecosystem of NIH resources. So now we have this backbone. It's anchored to 7,000 human traits with noodles of factors that are statistically rigorous. What's great about this is you can now hang additional knowledge graphs and orthogonal data sets to this. So what do I mean by that? Well, the world is changing. Knowledge graphs are everywhere. If any of you have heard them, it's like knowledge graph, knowledge graph. It's actually makes more meaning to me as I've learned more about it. So there's two major knowledge graphs other than ours within CFD. One is the C2M2 model knowledge graph, which we've already started to integrate into our model, and the data distillery. So these are two other knowledge graphs that connect CFD datasets through different means. So it builds a more holistic picture of the CFD ecosystem from a knowledge graph perspective. What's great is they will be interoperable and they'll be useful for many other resources, not just in the CFD. Another example of this is another CFD data resource center who's built their own KB, which is a biomarker KB. So again, we can attach to these, we can contribute to theirs. There's a two-way street here, which is really quite lovely. So beyond the CFD ecosystem, we can actually tap into an entirely new universe of knowledge graphs. So there's a really awesome collaboration that the NSF is sponsoring, which is called Proto-OKN. And the goals of that are to basically come together across NIH and NSF to initiate a publicly accessible, interconnected, open knowledge network. Very cool. So it's a massive web of semantic knowledge graphs that replaces these isolated silos and rigid databases in a queryable ecosystem. How do we do that? Well, we have to encode it in a certain way. And so I want to talk a little bit about that. If those of you who are not data scientists may not be as excited by this, but the architecture here is very straightforward. It's an idea that you can allow natural language query to an MPC client. And it basically, this is the underlying background, but it allows these knowledge graphs to be encoded in a Sparkle, enabled for Sparkle query. They're all encoded in RDF database and exposed to you. So that's the architecture that all of these resources in Proto-OKN are anchoring to. So this is not just the CFDE. There are many other resources. In fact, they have funding mechanisms to bring in knowledge graphs from across NIH resources. I actually don't know the number of how many, but I should get it. But it's all across biology. It's all across climate change. It's pretty impressive. So just to give you an example, once you encode your knowledge graph in this Sparkle query system, you can put a natural language query, example on the left here. It converts it to a Sparkle query and then reports to you back a natural language output. So that's kind of like the way that this mechanism works. But once we've done it in this format, we can immediately access a lot of other knowledge graphs, which is super cool. And that can be brought back to CFD and vice versa. So here's a nice example that Luca made, who's working on this in our team. He said, like, for example, if you took our knowledge graph and matched it against the PROT-KN knowledge graph, which is a knowledge graph produced by Uniprot, which is an incredibly valuable resource within our ecosystem as well, right? So you can do the same example query here. We can say, how do specific physical protein interactions explain the latent biological mechanisms in our model inferred for FADS2 gene? This just gives you an example. But you can do that with that's sort of the universe of proteins and genetics and genomics. But what about things like environmental stressors? There's a climate hub KG where you can actually ask a question like, which genes linked to respiratory mechanisms in our CFD graph are highly expressed in zip codes with poor air quality? So this is really cool. And they're going to build a UI for this that is tailored to different communities that will allow you to use natural language query to query across this entire knowledge graph. So we're involved in that, and I think that's really an exciting sort of additional direction for the CFD and the resource that we're doing. So the little summary of that, Luca is working on ways to get this schema finalized. And near term, that graph will be available for you to query in that Sparkle framework. In the future, we'll be integrating this more seamlessly with Proto-OKN, and you'll see some front ends for that that will also come back to the Knowledge Center. So that is actually it for me. I actually have some stuff about the incubator, but Mackenzie actually covered it. So with that, actually, I will stop and take questions or walk backwards through anything that was not clear.
Just a reminder, if you have any questions, please feel free to use the chat, the Q&A, or you can raise your hand, either way.
So what I'll do actually just real quick to give you guys a sense of what where you can find this. Let me make sure. Let's go here. So if you go to our Knowledge Center, it's right here, quarter way down the page. Start here and you type in your query. We have some examples here, but really type in whatever you want and see what you get back. And to be honest, any feedback is useful feedback. Even if it broke, it's great. I don't understand it. We're early in our documentation phase. One of the things we love to do, as we build stuff is to get it out there and kind of be, fearless in the fact that we'll put it out there, but also realize that like this is iterative and we want this to be useful for the community. So if it's not useful, please tell us, or if you'd like to see improvements or just like have a sit down with us. If you're like, I think this would be useful to me, but I'm not exactly sure how, we'd actually love that kind of interaction. That's really important to us. So we've worked with a couple collaborators in front of them and had them type in a query and kind of gotten a sense of what's coming back and what doesn't. And if it's something they expect. So that's really the important part. We want to know, do you expect to see any of these things? Does it give you like hints into insights into things you didn't know or want to know more about? And then a couple of the things just to note, this kind of approach, the sort of the context-based entrance into results and knowledge across scientific ecosystems is something we're bringing to our other portals as well. So you'll see these sort of types of interfaces percolating through the CMDKP, the Association to Function Knowledge portal, because I think that'll be really great for that space, because that's all about, finding genetic support for cross-human traits, and then what are the mechanisms, to understand the mechanism is critical to basically decide, if that's a target you want to pursue and whatnot. And then to some of our tissue portals around like the MATKP and things like that with the mammalian adipose tissue knowledge portal. So you'll see it in other places. And I think our goal there also is, if we do this right, you'll be sort of taken on a journey. It sounds a little silly, but taken on a journey through NIH resources that are relevant to you based on your context. And that's really the vision. So that's why this incubator matters. So this summer, when we get together with other NIH resources, there's like this whole universe of other resources out there. There's CFDE is its own thing, but then you have all the funded sites that work, I think it's like 51 of them under this PAR series for data repositories and knowledge bases. There's hundreds of those active resources every day that are producing data and results. That's a whole community by itself. You've got the NCAT's biomedical translator community, which is trying to build a huge sort of biomedical transfer across a host of resources in NIH. You've got Proto-OKN, I've mentioned. You've got the NCPI. These are all sort of different approaches to making data more useful. And I think our hope is that another way of making data more useful is scientific context, sort of operating as a layer on top of these. So not taking away from any of the beauty or the wonderfulness that these resources do, because that's their job and they're amazing at it. It's more about how do you bring people to those resources through their scientific context. Mackenzie, what's up?
We have a question, and
Beautiful.
this was submitted by an anonymous attendee. Do reveal outputs indicate what is novel versus what is known slash published, or clearly associated outputs with references versus first mark what are novel ideas generated?
Got it. Um, So that's a great point. No, we don't currently say novel versus known, and that's excellent. So I think this is, I'm wondering if DK is on because one of the things we're thinking about is like adding if it's already a published finding. That's a brilliant idea. So you get a PubMed link. Smart idea. So not yet. Another one is we do give you scores, so weighting scores, but that's based on the actual like linking in the pigeon graph and the statistical probability of that gene set or that mechanism being linked to that human phenotype. So that's all about statistics. But I think that's a really great idea to do novel versus known. I like that. We have to think about engineering that. If you have any ideas, send it our way. That's a really good idea. Other questions or comments?
You just want to say if there's anything else you'd like to see that we don't have, that's something we're also interested in. So if you think there's anything that would help other users, I'm sure they'll probably have similar feedback.
Mhm.
DK agrees it's a great idea.
Nice. So again, we have funding if you want to validate it, validate your findings. Tell us about it. Mackenzie, can you pop our help email too in there? That comes to like literally me and Mackenzie and a few other people, so it's not like it goes into a vacuum. Um, we are in early stages, but the CFDE is a very mature thing, and so we hope to make the CFDE data, more useful to you in our own little way, if you will. So any other questions? All right, have a go at it, and tell us how you feel, if it's useful to you. And if you're human genetics oriented or mechanism by like this is for you. Even though, we're talking about the common funded ecosystem, it really is anchored to human genetics. So I think that's really important to let you guys know about in terms of like caveats as you enter.
We actually have one other question.
Okay.
Can you do follow-up questions with CFD reveal output?
No, not yet. Even better idea. Wow, two ideas. Nice. I love that. DK, did you hear that? So like basically what you're saying, I think what you're saying is like you see a result, you're like, now tell me this, or now I want to see this. Is that kind of what you're saying? I'm asking you and you're not on mute, I'm realizing, but I'm assuming that's what you're saying. And so if that is, that's an excellent idea too. No, we haven't done that yet. That's brilliant. Cool. And if you're interested in provide like another thing, we are not shy. So if there's a better idea or a better way of doing something, or you'd like to add to our resource, we also welcome like development insight and help. So like those two feet, if you have an idea about implementation, that's also like important because implementers and data scientists are also users of CFD. And so that would be really valuable to have that insight if you are interested in developing those things. In fact, I think we have money, Mackenzie, for some challenges to actually do technical challenges as well, but that's for a future webinar.
That's good to know, yes.
All right.
Great. I don't see any other questions in the chat or Q&A. So I guess with that, I just want to say thank you, Noelle, for showcasing this new tool. As we have said, I just want to reiterate, please feel free to reach out at us at any point for any questions, comments, concerns, feedback. We are, like Noelle said, very receptive to all of that. And just a reminder to everyone that our next webinar is going to be April 24th, which will be in a month, and we look forward to seeing you all then. So thank you to everyone for joining us today, and thank you to Noelle for your presentation. Have a great rest of your Friday.
Take care, everyone.
Bye.